Bismillahirrahmanirrahim. Various methods for the measurement of hormone concentration in the blood are discussed in the video. So, the concentration of these hormones is very minute in the blood. They are about a billionth of a gram or a billionth of a milligram per ml, and so it is difficult to measure these concentration by usual chemical methods. So the methods that are usually used for the detection of these hormone in the blood are radioimmunoassays and the enzyme linked immunosorbent assay, that is also termed as ELISA. Radioimmunoassay is a method for the detection of hormones in the body. The first step in this method is detection of is the production of the antibody. A highly specific antibody is created to bind only the target hormone, for example, aldosterone or insulin. Then it is combined with the biological fluid or test fluid that contains the hormone, and a small quantity of the this purified standard hormone, that is radio labeled. So important condition is that there is not enough antibody. If there is not enough antibody to bind both the natural and reactive hormone completely, therefore the two forms of the hormone compete for the antibody binding sites. Both of the hormone, the natural one and the radioactive one, they compete for the antibody binding sites, and the more natural hormone in the sample, there is less radioactive hormone binds to the antibody. So the quantity of natural and radioactive hormone that binds is proportional to its concentration in assay fluid. Then the third step is separation and detection. After the equilibrium is reached, the antibody hormone complexes are physically separated from free hormone. The amount of radioactivity bound to the antibody is measured using a radiation counter, and it is interpreted as follows. If there is high radioactivity bound, there is low concentration of natural hormone or little competition for the binding, and if there is low radioactivity bound, that will be equal to, or the interpretation is that the high concentration of natural hormone is there. So there was a strong competition for the binding site. Then there is fourth step. The fourth step is the use of standard curve or quantification. A series of standard solution, a series of standard solution containing the known concentration known concentration of the natural untagged hormone are tested using the same method. The percentage of antibody bound radioactivity is plotted versus known hormone concentration to create a standard curve. It's like this that you can see over here, and then the radioactivity from the test sample is compared against this curve to calculate the unknown hormone concentration. This graph is illustrating a competitive radioimmunoassay used to measure the concentration of aldosterone in the test sample. So on the x-axis, the aldosterone concentration in the test sample in nanogram per deciliter. It is ranging from 2 to 128 on logarithmic scale, and on the y scale the percent of antibody bound with radioactive aldosterone ranging from 0 to 100% is given. Okay, and the curve shows an inverse relationship between the concentration of non-radioactive or test sample and the percentage binding of the radioactive aldosterone to the antibody. At low aldosterone concentration, like this 2 nanogram per deciliter, nearly 100% of the antibody is bound to the radioactive aldosterone. As the test sample aldosterone concentration increases, as this increases, it competes with the radioactive aldosterone for antibody binding, and this reduces the amount of this radioactive aldosterone that remains bound. At high concentration, that is at about 128 nanogram per deciliter, only a small fraction of the antibody is bound to the radioactive form. So this means that this is showing the concept of competitive binding. Unlabeled or cold aldosterone competes with the labeled radioactive aldosterone for a limited number of antibody binding site. This type of graph is used to calibrate the assay, so unknown sample concentration can be determined by comparing their binding percentage to standard curve. Radioimmunoassay like this one are used in endocrinology lab to measure the hormone levels such as aldosterone with high sensitivity even at a very low concentration. So the radioimmunoassay have got some advantages. They are ultra sensitive and they can detect nanogram to picogram of the hormone level, and they are also quantitative. So their precision typically occur within 10 to 15% error, and they are widely used for the hormone like aldosterone, cortisol, thyroxine, insulin, and growth hormone. The analogy that is used for to understand this concept is if you think like if you think of a competitive job interview, there are limited job slots that are antibody sites, and two candidates, the natural hormone and radioactive hormone, are competing. If natural candidates flood in, fewer radioactive candidates get the job. By counting how many radioactive ones got hired, you can estimate how many natural ones showed up. The enzyme-linked immunosorbent assay can be used for the detection of any protein, and the first step in this process is to prepare the plate. A 96 well plastic plate is used for this test, and each well is coated with an antibody, that is capture antibody. Here it is antibody 1. This is the capture antibody and it is coating the well, and then this antibody 1 is specific to the hormone that is to be tested. Then there is a sample addition. The patient sample or the standard hormone is then added to the well, and if the hormone is present, it binds to antibody 1 AB1 that has coated the well. So a second antibody is then used for the detection. The second antibody is specific for another site on the same hormone, that is antibody 2 in this picture, and then it is added. It binds to the capture hormone and forming a sandwich complex. So antibody 1 hormone and antibody 2 structure is formed. Then enzyme linked antibody is then used. This AB3 that is linked to the enzyme uh, is used, and this antibody recognizes AB2, and then a colorless substrate is added. This enzyme catalyzes this reaction, converting the substrate into colored or fluorescent product. The intensity of the color or fluorescence correlate directly with the hormone concentration. So non-hormone standard are run in parallel. A standard curve is generated and the optical signal from the sample is compared to the curve to calculate the hormone level. The key advantage of this process is that this is a non-radioactive test, and it is safer for routine lab work, and it is highly sensitive due to enzyme amplification process or reaction, and it is automatable and ideal for high through output testing, and it is cost effective and also accurate. So this is the diagrammatic presentation just to summarize or recapitulate this concept. This blue outline is the well that contains the specific substrate, and the initially it contains the coated antibodies that are for that are specific for that hormone. Then the hormone that is to be tested, it is then added, and when this hormone is added, this red hexagonal structure that labeled as H represent the target hormone. It binds to the antibody 1 that has layered this well, and then antibody 2 that is the detection antibody, it has got the specific site, another specific site or epitope on the hormone. This creates a sandwich complex AB1, AB2 and the hormone. This sandwich complex is then exposed to the AB3 that is linked with the enzyme. There is an enzyme, and this antibody 3 is specific for antibody 2, and this antibody 3 binds with the antibody 2, and it has got an enzyme, and when a substrate is added, the substrate S is added, this purple spheres labeled S, the there is enzymatic reaction by this enzyme E that catalyzes the conversion of substrate into these reddish spheres, the P or product. And the more product form, the more hormone was originally present in the sample. And the amount of colored or fluorescent product P that is formed is directly proportional to the amount of hormone H in the sample. This is the basis of quantifying the hormone level in the ELISA. So this was the basic principle of the enzyme linked immunosorbent assay. Now to compare both the test, radioimmunoassay and enzyme linked immunosorbent assay, the radioimmunoassay is it requires radioactive isotopes, while the ELISA requires only enzyme. So there is binding principle, that is competitive binding for radioimmunoassay, while antibody sandwich complex is needed for the ELISA. So signal interpretation, there is inverse relationship in the radioimmunoassay. That means more hormone will is equal to less binding of the radioactive level. While in ELISA, there is direct relationship. That means more hormone will lead to the development of more color or fluorescence, Radioimmunoassay is highly sensitive, while the sensitivity for ELISA is high, and the automation is limited in radioimmunoassay, while in ELISA it is easily automated in 96 well plate, and the safety uh, is quite a little compromised in the radio immunoassay because radioactive handling required, while in ELISA no radioactive substance is needed, only enzyme is needed. Thank you for watching this video. If you have any query, please write in the comment box.